Guys, today we're going to be discussing the ACDS system and the techniques and applications behind PCR detection of the ACDS systems and the use of the ACDS system in generating knockouts. Now, it's important to know what PCR is before discussing this, and it's essentially when you take a region of DNA that needs to be replicated, you denature, anneal, and elongate it, and then that results in a lot of copies of that specific section of DNA. There could be thousands, there could even be millions of copies, and that makes it very easy to then be analyzed with the amount of copies that there are. This paper is a good example of this. It's called an efficient field screening procedure for identifying transposons for constructing an ACDS-based insertional mutant library of rice. And essentially, they remove the DS tagging system in the rice, and they also insert a BASTA resistance gene. And then once the DS element is removed, as it says right here, the bar gene then becomes adjacent to the promoter, and this allows it to exhibit herbicide resistance. And what they did here was they were able to detect when the DS element was removed and then reinserted. And then it's written here, the P1 and P2 primer combination detected the excision, which was located at the 500 base pair band right here. This top row right here indicates when the DS was removed. And then the P3 and P4 primers detect if it was removed and then reinserted. And this is that lower band down here at 436 base pairs. And then along with this, they also did a combination of P5 and P2 primers. And then if these had a band at 600 base pairs, that means there was no excision, which is different than the 500 excision base pair line and the 436 reinsertion line. And this shows that if it's not removed, it's different from if it was removed, which is also different if it was removed and then reinserted. Along with the usefulness of PCR detection, there's also the ACDS system's ability to generate knockouts. This is shown very well in the work of Barbara McClintock. She was a Nobel laureate in physiology and medicine for her work in the cytogenetics of maize. It's important to know how her work kind of works. We previously stated it in the how ACDS works thing. And right here it shows how if a DS and the C locus are not in contact with each other, then the gene will operate as normally. And it'll produce the purple pigment here in the corn. And if the DS transposable element moves into or adjacent to the C locus, then it'll produce a mutant C locus, which doesn't produce the pigment. And then this is an example of how the knockout kind of comes into play. If the DS element is able to move into the C locus, it'll totally knock it out. And then Barbara McClintock goes further into detail in her paper, The Origin and Behavior of Mutable Loci in Maize. This is one of the things that helped her win the Nobel Prize in Physiology and Medicine. And then down here on page 7, the very top paragraph goes into detail of this. It says the insertion of chromatin composing DS the insertion of the chromatin composing DS adjacent to the C locus is responsible for complete inhibition of the action of the C locus, which is shown over here. As you can see, when it's adjacent or inserted into it, complete inhibition. And then she also further goes into detail on how if the DS is removed, the restoration of its action is due to the removal of the inhibiting DS chromatin, and then this allows for the normal expression to take place. So if the DS is then removed, it will no longer be a mutant C, it will go back to being the regular pigmented version. And then lastly, in this paragraph right after this, she goes on to say that there are many mutable loci that are in the genome of maize, and that there are many different ways you can create knockouts like this. Having a DS move into a different um, mutable locus will then knock out a different gene. And then, as well as this, we know that if the AC factor is there, as well as the C after the DS leaves, it'll produce a um, mixture of the two right here. And she explains this in this paragraph right here.